Welcome you. Today, we're doing a deep dive into, well, a really fascinating challenge in scientific imaging.
Mm.
It's something labs run into a lot, apparently. They've got these amazing microscope images, just mountains of them. Visual gold mines, really.
Absolutely, holding potential discoveries.
But here's the thing, how do you actually manage all that data? How do you find that one crucial image from, weeks ago?
Yeah, it's a problem that grows, right? The better the microscopes, the more data, and the harder it gets. It's
Exactly.
like having a huge library, but no card catalog. You know the info's in there somewhere.
But finding it, forget it.
Right, it's a real headache.
And that's exactly what we're digging into today. We got some insights from talking to someone, an intern actually, who's right there on the front lines trying to build a fix.
Okay.
So our mission here is to understand the current workflow, the, let's say, existing mess,
Mm.
look at this application they're working on called HOMERO, and figure out what difference it could actually make.
Sounds good. Where do we start? The current situation.
Yeah, let's start there. What's the day-to-day struggle like in this lab with these images?
Well, from what the intern described, it's basically individual hard drives. That's the main storage method.
Okay. So everyone just saves their images on their own drive.
Pretty much. Which sounds okay at first, maybe. But then, say a researcher needs an image from a specific experiment or a certain date.
Uh-oh.
Yeah, that's where it gets really bogged down.
I can just picture it. A drawer full of drives, labels maybe half useful,
Exactly.
plugging one in, waiting, searching, unplugging, trying the next one. That sounds painful.
It is. The intern really stressed how much time it eats up. You could spend like a huge chunk of your day just hunting.
Just looking for the data, not even analyzing it.
Right, and forget collaboration if someone else needs your image.
Nightmare.
It's awkward, inefficient. The lab doesn't even have a clear picture of all the data it actually possesses. It's just scattered.
Okay, that paints a very clear picture of the frustration. So that's the problem. Now, enter HOMERO, this potential solution. What's the basic idea?
So HOMERO is meant to be a centralized platform. The intern used a good analogy, like a lab-specific Google Drive or maybe even Google Photos, but, designed for these microscope images.
Okay, a central hub.
The core concept is one place, easy access, efficient management, store everything together.
Just getting it all in one place sounds like a massive improvement already.
It really is.
But it does more than just store them, right?
Yeah.
I think you mentioned something about searchable comments.
Exactly. And this is where it gets really interesting. A key feature they're working on is adding searchable metadata, comments, tags, whatever, to the images.
Okay.
The intern gave an example, like adding the comment cartouche to an image.
Cartouche. Okay, so how does adding a simple word like that change things?
Well, think about it. Say you need all images showing a specific cell structure or from a certain condition. If people tag their images with keywords like cartouche, then HOMERO search can just pull them all up instantly. Doesn't matter who took it or when or which microscope.
So you're not relying on fuzzy memory or file names anymore.
Exactly. You've
No.
got a dynamic, searchable database. It completely changes how you interact with the data.
That's brilliant. Turns static images into this active resource. Okay, HOMERO sounds great in theory, but getting it working. The intern mentioned an installation hurdle.
Ah, yes, the practical side. Setting up the server needed to run HOMERO. It was apparently quite a challenge.
How so?
The intern described it as very complicated, more like pure computer science work, not something a typical biologist would necessarily know how to do.
Right, managing servers is a whole different skill set than using, say, Excel or image software.
Definitely.
So how did the intern tackle that if it wasn't their main area?
Resourcefulness, basically.
Yeah.
They mentioned relying heavily on online guides, documentation tutorials, yeah, the usual tech problem-solving approach.
Okay. Googling their way through server setup.
Yeah.
Been there.
Yeah, but it does highlight a potential barrier, right?
Mhm.
If other labs wanted to use something like this, they might hit the same wall if they don't have someone with those specific tech skills.
True. You shouldn't need to be a sysadmin just to organize your research data. So HOMERO's still being tested. It's early days. What's next for it? User feedback sounds key.
Absolutely crucial. The intern actually has meetings set up with colleagues, the researchers who will be using it every day.
Ah.
The goal is to really nail down their specific needs, their current analysis workflows. How do they work with images?
That makes sense. Build it for the users.
Yeah, because you can have the fanciest tech, but if it doesn't fit how people actually work, they just won't use it.
So true. So beyond storage and search, what else are they hoping Homero can do? Any specific features?
One really interesting idea being explored is getting Homero to automatically detect which microscope an image came from.
Oh, why is that important?
Well, apparently different microscope brands like Zeiss, Nikon, the intern mentioned those, they often use their own proprietary image file formats.
Ah, so not like a standard JPEG or TIFF always.
Exactly. And these special formats often need specific software, usually from the manufacturer, just to open and analyze them properly.
Okay, I see. So you've got these different silos based on the equipment.
Right. So the vision for OMERO is it not only stores the image, but it knows it's, say, a Zeiss image.
Mm-hmm.
And then it could potentially offer to open that image directly in the correct Zeiss software for analysis.
Oh, that's clever. Seamless integration.
Yeah, it would really streamline the whole process, from finding the image in Homero to actually working on it in this specialized tool.
Like a central command center that connects you to the right tools automatically. The intern compared it to the Google Suite, right? Drive connecting to docs or photos.
That was the analogy, yeah. A Homero is the hub for the image data, intelligently linking out to the specialized analysis tools, making the whole workflow feel connected.
Okay, this intern sounds pretty central to all this. What's their specific role? What are they actually producing?
Well, two main things. First, as we said, they tackled that tricky server setup.
Right, the installation hurdle.
And second, they're creating user guides, manuals basically, to help their colleagues get started with Homero.
Makes sense. Got to teach people how to use the new tool.
Yep. They were originally in French, apparently, but are being translated into English now for wider use in the lab.
Good call. And what's in that first guide? The basics.
Pretty much how to actually upload your data, connecting your hard drive, finding the folders in the Homero interface,
Okay.
selecting what you want to import. They even mentioned a little shortcut, like using Ctrl+Click to select multiple folders at once.
Ah, the little tips that make a difference.
Those always help.
So you select your folders. How does Homero organize them?
It uses a project-based system. So the example given was, like, if you did experiments on Monday, you'd create a Monday project in Homero
Got it.
and import Monday's images into that. Then Tuesday's images go into a Tuesday project and so on. It gives you that chronological or experiment-based structure.
Seems intuitive. Easy to keep track of things that way.
Mm-hmm.
So the intern's goal with these guides and maybe a presentation is just to get everyone comfortable using it.
Exactly. Get them onboarded, show them how it works, how it can make their lives easier than juggling all those hard drives. Drive adoption, basically.
Right. Shift them away from the old clunky way towards this new, hopefully better system.
That's the plan.
Now, you mentioned the intern wasn't only working on Homero. There was something else. Deep learning.
Yeah, a secondary task.
Yeah.
They're also involved in trying to deploy some deep learning code written by someone else, a colleague named Jules.
Okay.
And they're trying to get it running on a specific platform called RiceSide.
RiceSide. And why use RiceSide? What's the advantage over just running it on a normal computer?
Performance, mostly.
Yeah.
RiceSide is apparently a platform with much more computational power.
Ah, for heavy-duty processing.
Right. Deep learning, train these complex AI models, it often needs a lot of grunt. Specialized hardware, maybe GPUs. RiceSide offers that kind of environment. Faster training, faster results.
So it's like a high-performance computing resource. Makes sense for AI stuff. But there is a snag.
Yeah, a bit of a roadblock. They're currently waiting for account access to RiceSide.
Bureaucracy.
Happens all the time in research, right?
Yeah.
Waiting for permissions, access, infrastructure to be ready. So that part's on hold for now.
Understood. So stepping back a bit, what's the intern's own background? Why are they doing this specific internship?
They're a third-year licensed student, so an undergraduate. It's a 2-month practical internship focused specifically on data management.
Okay.
And they apparently sought this out because they're interested in a career in data analysis.
Yeah.
So this is perfect hands-on experience for them.
Oh, fantastic. So it directly lines up with their goals. Building a tool like Homero, getting exposure to platforms like RiceSide. That's great CV material.
Absolutely. It's real-world experience, applying what they've learned, picking up practical skills, bridges that gap between academic study and actual research challenges. A really valuable opportunity.
Definitely. Okay, so let's wrap this up. As we finish this deep dive, what are the main takeaways for you, the learner, listening in?
Well, I think the big one is just how critical and often tricky managing scientific data really is. You know, the science might be cutting edge, but just handling the data it produces can be a real bottleneck. Homero is like this focused effort to fix that for microscopy. And the fact that it's being designed with the researchers
Yeah.
based on their needs, that feels like a good model. Maybe tools like this could even, spark new ideas just by making data easier to find and share.
Yeah, and for me, the aha moment was really grasping the problem of all those different file formats from different microscopes.
Right.
It's not just storage, it's about bridging those technical gaps, making the data truly usable in the researcher's actual workflow. Think of the time saved. Less searching, more analyzing.
Potentially speeding up discovery, yeah.
Exactly.
And that leads to a final thought, maybe, for you, the learner, to ponder. What about other scientific fields? Where else is data management this kind of challenge?
Good question.
How can seemingly simple organizational tools, like OMERO aims to be, unlock bigger research potential just by making data easier to handle? And are there lessons from this specific example, this intern tackling image chaos, that apply elsewhere?
Lots to think about there, how small tools can have a big impact. Thanks for exploring that with us today.